So the cross which involves two pairs of contrasting characters is called as dihybrid cross. It's called as dihybrid cross. That means here we have to take two pairs of contrasting characters. For example, sheet shape. It has two varieties, round and wrinkled. And seed color, yellow and green. For corresponding characters or traits, the genes are like this. Capital R, capital R, capital Y, and small r, small y, small y. So this will be your round yellow parent. This will be your wrinkled green. Similar is the mechanism of the methodology of hybridization. Then gametes. So this will be your F1. The F1 will be also round and yellow. Now we will perform the selfing of F1 generation, producing 4 gametes. Identical gametes as it is selfing. This will be for male and this will be for female. So parentals at the end, F1 in the diagonal, then so these are the genotypes. And what will be the phenotypes? Round yellow, round yellow, round yellow. 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 round yellow. Then round green. Look at the round green. This one is round green, 2, and this round green. Then we will have wrinkled yellow. What will be their population percentage? Wrinkled yellow. So this one, wrinkled yellow, 3, and only one will be parental, wrinkled green. So the phenotypic ratio is 9:3:3:1. And if carefully you will find out all the genotypic ratio, then it will be 1:2:2:2:1:2:1:2:1. This will be the genotypic ratio. 2 2 4 4 8 1 9 10 2 12 13 14 15 16 total होची 16. एठी देखो टोटल केते अछि 4 टा 4 into 4. So here it is 16. एठी 9 3 12 15 1 16 16. So correct. सब गुडा जदी 16 हे गला, then you will check that your checkerboard is completely correct. 15 की 14 हला माने कोडे भूल कर देजा. So This will be your, the phenotypic ratio will be 9:3, 3:1, and the genotypic ratio will be 1:2:2, 4:1:2, 1:2:1. So मन रखिबार इजी होची 1 2 2 2 2 4 1 2 1 2 1 एवला मन रखिबो नले गोटे गोटे बाहर करिवो यदि एटा मन रखिला ताले एटा को गोटे गोटे बाहर करिया दरकार तो एग्जाम रे किंतु phenotypic ratio as well as genotypic ratio लिखिबो. So test cross भी कहइतली. So what will be the test cross? This will be the test cross. F1 hybrid crossed with the recessive parent. So again 4 types of gametes. But here it is only one type of gamete. So your round yellow, round green, wrinkled yellow, wrinkled green. 1:2, 1:1, 1:1. So this will be the test cross ratio 1:1, 1:1. So this is about your dihybrid cross. Dihybrid cross paved the way for law of independent assortment. Independent assortment. It is also known as the second law of inheritance. Known as the second law of inheritance. It is applicable to minimum, at least there must be 2 contrasting factors. Otherwise, for single it is not applicable. As there is independent assortment of gametes with the formation of the individuals, so or the phenotypes. That's why it is the law of inheritance. So next part is incomplete dominance. Incomplete dominance. Actually, the monohybrid cross and dihybrid cross prove that when 2 factors or more than 2 factors are present together, one of the factor expresses completely. So this individual is completely tall. Shows one of the parent. What a parent show करे. Similarly, when 2 factors are taken into consideration, it is round yellow. So this shows that always a single dominant parent's trait will be exhibited. That is known as principle of dominance. Principle of dominance. Only one trait or one individual's characters will be reflected or exhibited. That is called as principle of dominance, or it is also the absolute dominance. Absolute dominance. Why? काहे ना टॉल और डार्क मझिरे आमे किछि पाइनु बा राउंड और येलो किछि मझिया मझि आमे रिजल्ट पाइनु. एक्जेक्टली जेउटा राउंड येलो पैरेंट रो ये मध्ये से राउंड येलो ये भी टॉल पैरेंट F1 रे मिलला. So that is absolute dominance. But in nature actually complete dominance is not always observed. Or in example, snapdragon. Snapdragon. Antirrhinum majus. This is the scientific name. In Antirrhinum majus or snapdragon, there are populations of 2 flowers. One is red, another one is white. So these R genes are supposed to contain pigmentation. तेणु R gene pigment बा red pigment formation करूची. यदि parent generation में cross करिबा, gametes will be like this. F1. हम previously जाने जो complete dominance, so we will expect red flowers. कौन सी गोटिए parent को show करिबो. But in nature, pink flowers appear. So this is a deviation of principle of dominance. Further यदि selfing करिबा 3:1 ratio आसीबा दरकार. But when it was studied, it was 1:2:1 genotypic ratio. Simultaneously, the phenotypic ratio was also the same but with a different intermediate phenotype. मझिरे गोटे pink color बाहरिला. It is due to incomplete dominance. So how incomplete dominance is explained? Suppose your R gene, it will produce an mRNA, and that mRNA will produce pigment protein. It will produce a pigment protein. When RR full dose, so red. When smaller, no mRNA, so no pigment theory होनी, so that will lead to white, किसी pigment नहीं. But when this will take, this is half dose, आधा dose रे अच्छी, so it will be pink. So this is your incomplete dominance, explained as such. Red, pink, white. But this is the phenotypic ratio is 1:2:1. Genotypic ratio RR, capital R, small r, that is also 1:2:1. तेणु काउंसिल गोटे वर्ष पढ़ी क्वेश्चन जे if the genotypic and phenotypic ratio is same, then what is the relation between the genes or the traits? That is incomplete dominance. So you have to remember the genotypic and phenotypic ratio is same in case of incomplete dominance, but in complete dominance the phenotypic ratio is 3:1, but the genotypic ratio is 1:2:1. It is the difference between incomplete dominance and complete dominance. So next part is, so after incomplete dominance, another kind of diluted form of dominance is observed, that is co-dominance. Co means equally represented. Here co means equally represented. कंप्लीट डोमिनेंस रे गोटे पेरेंट एक्सप्रेस कला, इनकम्प्लीट डोमिनेंस रे दीटा पेरेंट रो मझिया मझी फैक्टर असला, को डोमिनेंस रे उभय पेरेंट को कैरेक्टर माने या कथा भी रहबो ता कथा भी रहबो. तार एग्जांपल होची तमरो AB ब्लड ग्रुप, AB blood group in human beings. Blood group विषय जानिछो human beings रे A, B, AB, O. मोरे पॉजिटिव नेगेटिव RH फैक्टर कथा कोन जे इनहेरिटेंस रे RH फैक्टर कथा मेंशन करूनी. सो दिस AB ता पूर्व जानिबा जे ब्लड ग्रुप डिसाइड केमती हुए आ ब्लड ग्रुप कोन. This is your RBC. This is a biconcave RBC. RBC surface रे केते गुड़िए ओलिगोफोरिन और एंटीजेन बॉडीज थांती that forms the blood group. ता पाई स्पेसिफिक एलिली मध्य अछि. जदि एलिली एमिति रहिला, जेहेतु डिप्लॉयड ऑर्गेनिज्म एमिति रहि पारे और एमिति रहि पारे, से केस रे से इंडिविजुअल ब्लड ग्रुप A आसीबो, as the allele A is dominant over O. IB ये एभल भी पारे, it will be B, as the B blood group allele is also dominant over the O allele. But now we'll come to our point. AB, IA, IB, IA हुई, IA, IB. So this will be your AB blood group. Why? Because IA allele is co-dominant with B allele. So IA, IB will equally represent. So this type of persons, RBCs, will contain the antigens but no antibodies. So two antigens. So this will be your AB blood group. What about O? This has to be homozygous recessive. A case रही केवल O हबो. नहीं ना O टा होची recessive. But मन रखिता population of O is highest than any other alleles. Population of O सबुठु अधिका. So we will next part will be our multiple allelism. Multiple allelism. So before going to multiple allelism, we know what is an allele. It is the alternative form of a gene. Alternative form of a gene sharing a common locus. Sharing a common locus on chromosome R. Alternative form of a gene sharing a common locus. So, this is a chromosome with two distinct arms. तार फिक्स्ड कोड फिजिकल पोजीशन जोठी एलीली थाए, it is called as a locus. Suppose allele is A. So this is your allele. सेमती homologous counterpart रे suppose example हमें देइछु tall rock capital T small t. These two are different alleles किंतु सही गोटी character होची हमर height. Character is our height. And our height. Locus is same, capital T small t. When there are more than two alleles for the character, then that sort of event is called as multiple allelism. Suppose there are three alleles for any hypothetical character, A dash, small a. In this case, we will say it is exhibiting multiple allelism. Or in the same manner, जोटा कि blood group रे भी पढ़ी दिले, blood group रे IA is there, IB is there, and a third character is IO. So your ABO blood group again exhibits multiple allelism. ABO blood group. Multiple allelism. So there are certain characteristic features of multiple allelism. So characteristic features of multiple allelism. This is very important. तुमको एटा ही परीक्षा रे लिखिबो अछि. एगुडा न लिखले चलिबो. एटा बुझेबा पई कहली. लिखिबा पहिले first point. What are the characteristic features of multiple alleles? A diploid organism contains maximum two alleles. Each chromosome, maximum two alleles. Gametes contain only one allele. कारण गैमेट जितना तैयारी हो सेपरेट हो हैप्लॉइड गोटे गोटे, but the entire population will have all the alleles. सब एलीली रही जीवो. तुम जे ह्यूमन पॉपुलेशन को स्टडी करिवो, ए थिबे, बी थिबे, ए बी थिबे, ओ थिबे with positive and negative. So these are the basic characters. Next, share a common locus. They share a common locus. ए जोडा कहली ये जोटी थिबो कैपिटल टी पई स्मॉल टी भी सही जगह रे थिबो, आ को अलग जगह न थाए, एडी तळे मध्ये न थिबो, कि आउ एठी कोठी बा ए को आर्म रे न थिबो, केवल ठीक ता सीधा रे सही होमोलोगस क्रोमोसोम रे थिबो. Next is there is no crossing over between members of multiple alleles. They may exhibit absolute dominance, incomplete dominance, और जोटा एबे कहली कोडोमिनेंस. कोनोसी प्रकार बिहेवियर देखे पारे किछि ता माने नै जे समस्त भितर गोटी प्रकार थुव बहुत गुडे एलिमेंट छथि. सपोज ए या पई होची तमरो डोमिनेंट, बट आ पई होची कोडोमिनेंट, बा ये आ पई इनकम्प्लीटली डोमिनेंट, किछि भी प्रॉपर्टी बिहेवियर शो करि पारंती. सो दे मे एक्सिबिट डोमिनेंस, इनकम्प्लीट डोमिनेंस एंड कोडोमिनेंस. देयर इज ऑलवेज ए वाइल्ड एलील जोटा सबुठु फास्ट आसीथिबो बा पॉपुलेशन रे सबुठु अधिका थिबो दैट इज अ वाइल्ड एलील and its mutation produce mutants. Mutations means genetic structure बा तमरो DNA रो sequence रे कौन सी alternative changes to mutant theory हेबे. Next is very important is the examples. यति कि लिखले भी full mark मिलिबो नहीं. So what are the examples? Eye color in fruit fly or Drosophila melanogaster. Drosophila melanogaster. Next is ABO blood group. Next is ABO blood group. Number 3 is S allele based sexual incompatibility. S allele based sexual incompatibility. It has 4 such alleles: S1, S2, S3 and S4. So next is chromosome theory of inheritance. Before going to chromosome theory of inheritance, so what is Mendelian inheritance? So first we must know what is Mendelian inheritance. जोटा मेंडल प्रपोज करते Mendelian inheritance. Number 1, Mendel coined the term factor or elementi which determines the characters. Factor or elementi which determine the characters. Next, they exist in pairs or they remain in pairs. They segregate independently into gametes. Law of segregation. They again unite in the hybrid. On selfing, they show independent assortment. So this is your Mendelian inheritance. And it is also according to classical genetics, as there was no development of tools and techniques. So it was classical genetics concepts of Mendelian inheritance, that the factors are responsible for the characters, they exist in pairs, they segregate independently into gametes and again unite in the hybrid and exhibit independent assortment. It was Walter Sutton and Theodore Boveri. Walter Sutton and Theodore Boveri in 1905 studied chromosomal behaviors under microscope, which is called chromosome theory of inheritance. So what is this? First one is there is no factor, rather chromosomes. The chromosomes occur in pairs and they determine the characters. It is called as the physical basis of heredity. The physical basis of heredity. They remain in pairs. It is also same. They remain in pairs. They segregate, show law of segregation, unite in hybrid, and finally show independent assortment. এটা তোমার short note আসিব 2.5 মার্ক সো ইউ হ্যাভ টু কম্পেয়ার মেন্ডেলিয়ান ইনহেরিটেন্স অফ देयर इज अ पैरेललिज्म মানে সমান্তরাল ব্যবহার যেটা উপতি প্যারালাল বিহেভিয়ার অফ মেন্ডেলিয়ান ফ্যাক্টরস উইথ দ্যাট অফ ক্রোমোসোমস ফার্স্ট they carry the physical basis of heredity or they carry the determine the characters they remain in pairs they segregate independently they unite in the hybrid again show independent assortment पूरा completely जहाँ जहाँ मेंडेलियन फैक्टर्स शो करते एटी क्रोमोसोम से बिहेवियर शो करती दैट्स व्हाई इट इज कॉल्ड एज chromosome theory of inheritance it's completely parallelism with the Mendelian inheritance. Your next part is polygenic inheritance. Polygenic inheritance. So it is similarly we have already studied monogenic inheritance or Mendelian qualitative inheritance. Monogenic inheritance. This one is polygenic inheritance. Monogenic inheritance and polygenic inheritance. So what do you mean by monogenic inheritance? Single gene determines the trait like your tall and dwarf. The traits are qualitatively studied. That means we can say tall or dwarf, but we cannot say tall how much tall and how much dwarf. Tall टा कितने डेंगा, dwarf टा कितने गेड़ा, तार कौन सी measurement नहीं. So it is simply a qualitative study. Next, discontinuity was observed. Discontinuous traits. डेंगा और गेड़ा में बहुत difference है. So we can say it is a discontinuous trait. And no impact of environment. No impact of environment. On the other hand, what is a polygenic inheritance? It is controlled by a number of genes. It is controlled by a number of genes. किसी माने नहीं जैसे जीन गुड़ा से गोटी क्रोमोसोम थी, अलग-अलग क्रोमोसोम भी दिखाई करती. So a number of genes present on different loci or different chromosomes. किसी माने नहीं जैसे गोटी क्रोमोसोम रही सब उड़ा ही थी. दीटा एठी थी चारटा आउ क्रोमोसोम थी किंतु समस्त मिसी की गोटी कैरेक्टर को डिफाइन करूथुवे बा एग्जिबिट करूथुवे. A number of genes. Next, each dominant gene has additive effect on trait. जेते जेते डोमिनेंट जीन हमें ऐड करी बा जेते जेते डोमिनेंट जीन थिबा से ऑर्गेनिज्म जीनोटाइप रे तार फिनोटाइप सेते प्रोनाउंस्ड हबो. हियर इट इज अ क्वांटिटेटिव स्टडी ऑफ द ट्रेट्स. डेंगा हेले तार मेजरमेंट दरकार केते डेंगा ग्रेडेशन केते डेंगा मानकर पुनर रेंज थिबा दरकार. सो देयर इज ए क्वांटिटेटिव स्टडी. देयर इज अ कंटीन्यूअस वेरिएशन. एग्जांपल देले बुझी परिबो. देयर इज अ continuous variation. That is a continuous variation of the traits. माने डेंगा रू गेड़ा बा गेड़ा रू डेंगा जेमिति आसले बड़े continuous variation observe होतबो. controlled by environment. It is controlled by environment. So this is the basic difference between your monogenic inheritance and polygenic inheritance, or it is also known as your quantitative inheritance. Quantitative inheritance. This is your quantitative inheritance. A very important example is given for quantitative inheritance. It is the human skin color or complexion. Human skin color. It is controlled by 3 pair of genes. Three pair of genes, and if we will find out the phenotypes, they are found in this 1:6 is to 15 is to 20 is to 15 is to 6 is to 1. So 1 is to 6 is to 15 is to 20 is to 15 is to 6 is to 1. This will be equal to your 64. Tri-hybrid cross जदि दबा सेठी से कोन हबो 4 into 3 64 outcomes will come, or the 64 individuals will be studied. So एठी हम कोन लिखिबा very fairly light सबटु कम population, fairly light. It is simply fair. This one is intermediate or Asian Caucasoids. Intermediate. This one is fairly dark. This one will be your dark, and this is very very dark. That is negrito or negroid. So this is the 7 types of phenotypes. 7 types of phenotypes. 7 प्रकार रो phenotype polygenic inheritance रे देखा जाय. और सबुठ important last part controlled by environment. जेते polygenic trait हो जदि जीन जहां भी था हो, if environment is not supplemented or proper environment पाइबो नहीं, exact character show कर परिबो नहीं, so there will be a dilution in that character. तो जदि proper condition or proper environment no पाइबो, it will not go to that attainment of that trait. That is the certified controlled by environment. Most of the characters are controlled by polygenic trait or polygenic inheritance. ये गोटे वहाँ सीरे control हुए नहीं, तो ये polygenic inheritance. बहुत बड़े example है. तो actually जो ना Mendelian height से इटा मध्य वो polygenic inheritance. Your height, color, size of fruit, all are environmentally or hormonally controlled along with the genes. So this is all about your polygenic inheritance. So your next part is pleiotropy. It is the phenomenon in which one gene influences a number of phenotypes. A number of phenotypes. Phenotype 1, phenotype 2, phenotype 3. Example रे बुझी परिबो. For example, हमर काउंसिल रे गोटी एग्जांपल ही अछि. नले प्लियोट्रोपी बहुत इवेंट अछंती. किंतु काउंसिल रे गोटी अछि PKU. Phenyl ketonuria. So PKU. It's a disorder. It is a metabolic disorder in which exactly कौन हुए phenylalanine गोटे amino acid ये convert होगा tyrosine by enzyme phenylalanine hydroxylase. Phenylalanine hydroxylase. Tyrosine is the precursor of many important neurotransmitter or hormones. Epinephrine, norepinephrine, and dopamine. Adrenaline. This is your adrenaline, and this one is noradrenaline. Adrenaline and noradrenaline, dopamine. So देखो एठी जो जीन टा ए एंजाइम क्लियर करूथबो से इनडायरेक्टली एक्चुअली तीनटा प्रोडक्ट फॉर्मेशन रे हेल्प कर जिबा तीनटा अलग अलग फिनोटाइप या काम अलग या काम अलग या काम अलग तो तीनटा अलग फिनोटाइप को गोटिए जीन ही कंट्रोल करूची यदि यार कोनसी जीन रा abnormality हैला so this synthesis will be blocked and the individual will be converted phenylalanine into pyruvic acid formation हेकी तार urine रे disorder आसीबा accumulation of homogenistic acid formation होबो so this is epinephrine norepinephrine and dopamine एटा तीनटा को फिनोटाइप गोटिए जीन रु कंट्रोल होचंती that is called as your pleiotropy so why pleiotropy arise it is due to the interrelationship of metabolism जेतिको मेटाबॉलिज्म अछि माने इंटररिलेटेड दैट्स व्हाई दिस प्रॉब्लम अराइजेस बा गोटिए जीन चाहिए बहुत गुड़िए यदि गोटे की एंजाइम गोटे रेगुलेटरी एंजाइम को कंट्रोल करथबो तो इट कैन इजीली रेगुलेट many phenotypic processes that is called as your pleiotropy next part is your linkage and crossing over linkage and crossing over so first you must know what is a linkage suppose गोटे क्रोमोसोम रो गोटिए आर्म रे दुईटा जीन ए आउ बी पाखा पाखी अछंती so जेतबेले का क्रॉसिंग ओवर हो so there is a more chance that both of the genes go simultaneously दी जण जाका एका बेले बोले ए अलगा ही परिबो नहीं केबे भी बी थारु सपोज एठी मध्य ए आउ बी पाखा पाखी जेन ए बी पूरा पूरा चली आसबे ए कैपिटल बी पूरा सब चली जिवो किंतु हमें ए आउ बी को अलगा कॉम्बिनेशन माने ए भलिया कॉम्बिनेशन हमें पाइबा नहीं देखि कि बुझी परछो कैपिटल ए स्मॉल बी हमें पाइबा नहीं स्मॉल ए कैपिटल बी पाइबा नहीं if they are present very close to each other this phenomenon is called as linkage तर मन पकाओ यदि एई जिंसटी हबो बा एई लिंकेज हला हमें कैपिटल आर कैपिटल वाई जेतबेले क्रॉस करिथिले कैपिटल आर कैपिटल वाई यदि एमाने लिंक देतांदे प्रॉब्लम कोन हितांदा ओनली these two types of gamete will be formed किंतु आमे चारिटा गैमेट पाइथिले तेनु independent assortment होबो नाही so linkage is a deviation or in linked genes do not exhibit independent assortment show करि परिबे नाही जीन माने restricted ताको मूवमेंट रे ये ताको ता संगे ही नेकी पड़ेबो and these genes are called as linked genes these genes are called as linked genes so linkage study के लिए भाई उन्हें दीटा एक्सपेरिमेंट अछि that is called as coupling and repulsion coupling and repulsion in Lathyrus odoratus two experiments first is coupling means coupled or sangre rahla so blue long stamen red round stamen से crossing होगा blue flower with long pollen red flower with round pollen blue long red round when this will be crossed this will be your blue long F1 यार वर्तमान टेस्ट क्रॉस करा जीवो टेस्ट क्रॉस रे ही रेशियोटा बाहर टेस्ट क्रॉस रे we have chance that there will be blue long there will be blue round there may be red long or the parental red round ए भया आसीबा दरकार किन जेतबे एक्सपेरिमेंटली प्रत्येक प्लांट को मंजी लगे कि पुणी गछटा उठिला से पाइले एई रेशियो रे and not that 1:1:1:1 एई रेशियो रे मिलिला ता माने ब्लू लॉन्ग एठी जो बी सहित एल टा कपल हेकी थिला से एंड जाए लिंक हेकी रहिला एठी जे रेड सहित राउंड थिला सी भी मध्य एंड पर्यंत लिंक हेकी रहिला सो दिस इज कपलिंग फेज देन रिपल्शन फेज रिपल्शन रे कोन हेची ना ब्लू राउंड एंड रेड लॉन्ग that means repulsion है कि वो अलग रह जाती blue round red long gametes will be like this repair ହେଇକି ଅଛନ୍ତି but constant will be this एटा same ରହିବ this one will remain same blue long it has again has 4 chances blue long blue round red long red round एभଳି भाବରେ ରହିବ blue long blue round red long red round 7:1:1:70 बारला कपलिंग फेज रे रिपल्शन रे कोन देखाबो ना blue round आ red long एमाने सबुठु अधिकार रहले सो रिपल्शन फेज भी इनिशियली धरा होनता जेत पार्ट ऑफ लिंकेज बट लेटर ऑन ऑब्जर्वेशंस लेट दैट it is also a part of linkage कहीं ना एठी मध्य देखो इंडिपेंडेंट असॉर्टमेंट रो रिजल्ट हमें पाउनु 1:1:1:1 रो रिजल्ट मिलु नाही सो दिस इज योर कंप्लीट लिंकेज आज सेटिंग